And since it's so dangerous, it's the Maine Department of Marine Resources to keep the public safe, and they do that by closing down shell fisheries. And every week a shell fishery is closed down, it loses $3 million just on the coast of Maine, so it's really important to be able to concisely know when it's going to happen and prepare for these harmful algal blooms, which are called HABs. Once Alexandrium is no longer an issue and the bloom has subsided, the cells group together on the nepheloid layer, or the bottom layer of the ocean, and create a cyst bed. And in a cyst bed, Alexandrium waits for suitable conditions such as nutrient-rich water, high dissolved oxygen levels, and warm water to bloom again. And it can actually remain dormant for up to 100 years. Maine has two major cyst beds, which you can see by the Bay of Fundy cyst bed in the blue arrow, and the Casco Bay/Penobscot cyst bed by the white arrow. And you can also notice that on this map, most of the coast is covered by a red line, and this line represents all the areas along the coast that have had their shell fisheries shut down since 2005. So it just shows how widespread this problem is along New England's coast. You can also notice that there's black arrows, and this is Maine's currents. And Maine has very strong and distinct currents, which moves plankton around a lot. And so one of the many reasons why it's important to be able to know the genetics of Alexandrium is to know where each cyst bed is, where their cells are going.
By looking at Alexandrium fundiense's genetics, we could be able to see a population divergence within the Gulf of Maine. If there is a population divergence of A. fundiense within the Gulf of Maine, this could mean that these cells could have different bloom triggers, meaning the pH in the water temperature, and salinity. And these bloom triggers are actually what causes harmful algal blooms. And so by having different bloom triggers, these A. fundiense could have different effects on the humans consuming the cells within the bivalves. If they are different populations, they can produce different toxins, like Betsy talked about earlier. A. fundiense produces a toxin called saxitoxin. However, if there were different populations, they could produce a different kind of toxin, therefore harming humans in a different way.
Through all of our research, we got a research question that would reflect well on our research and all of our questions that we've been asking. And our research question would be, is there a genetic difference within Alexandrium fundiense between the two cyst beds in the Gulf of Maine?
From that question, we formed a hypothesis, which is if Alexandrium fundiense comes from different cyst beds, then the genetic codes of the ITS1 gene will be different, therefore indicating a population divergence.
Although we started this project a few weeks ago, really data collection started years ago when volunteers at Maine DMR started collecting samples from 4 different sites. The Orr's Island site, which is the green arrow, or green star, and Phippsburg, which is the blue star, both of which correspond to the Casco Bay/Penobscot Bay cyst bed, and then Jonesport, the red star, and Gleason's Cove, the purple star, both of which correspond to the Bay of Fundy cyst bed. The volunteers collected 115 samples starting June 22nd, 2010 and ending September 2nd, 2014. Of those 115 samples, 29 were found to contain over 100 cells per liter of Alexandrium fundiense. Our work started in the lab, where we first attempted to isolate individual cells of Alexandrium. However, this was complicated by the age of the samples and the fact that they had been added legal solution to attempt to preserve them. All in all, we didn't completely isolate it, but we moved forward to whole genome amplification. We replicated genes in order to have more genetic material that we could work with. We did this by adding buffers to create the right condition and enzymes to replicate. Then we put it in the thermocycler for 16 hours. Our last step was polymerase chain reaction, which was replication targeted on a specific region of the genome. We again added buffers and enzymes, but this time we also added primers to target the ITS1 gene. We chose ITS1 because of its prominence as a phylogenetic marker, or a marker that indicates a certain species at indicates what we're looking at as a species and population level. Lastly, we sent it off to Yale Genetic Sequencing Lab for Sanger sequencing.
Our data came back as an electropherogram from Yale Sequencing Labs. And an electropherogram is a diagram that shows the specific genetic sequencing of the selected DNA cells that we chose to analyze. In this case, we had A. fundiense cells. However, Meredith just spoke about, these cells weren't completely isolated due to their unnatural preservation and their old age, since they were first collected in 2010. This meant that the electropherogram processing wasn't able to solely pick up on the Alexandrium cells, and they also picked up, as you can see in the electropherogram image above, different types of organisms that were in the isolated cell tubes when it was processed. This caused the electropherogram to display overlapping templates, which means it is displaying overlapping genetic sequences of these various amounts of cells. Since we could not see a clear population divergence of the A. fundyense cells within the Gulf of Maine, we decided to look at A. fundyense globally instead. We chose 6 globally different samples of A. fundyense from around the world. These samples were collected off of a library of genetic sequences called GenBank, which is available to the public, which holds the specific DNA sequences of every cell that is in that program. We then put these 6 samples into a phylogenetic tree to see their relation. This phylogenetic tree, as you can see above, shows 2 specific groupings, one in the Pacific coast and one in the Atlantic coast. And you can notice that the 3 samples in the Pacific coast are more closely related to each other than the ones in the Atlantic coast. This means that even though that A. fundyense cells are different, they can also be similar. However, you may notice that none of these samples are 100% genetically similar to each other, which can also infer that no matter how close the sample is, they can still be genetically different. We can make a hypothesis out of this phylogenetic tree that location plays a huge part in how diverse A. fundyense cells are globally on a global level.
Going back to the Gulf of Maine, we used our samples that we received from the Maine DMR to make a chart of the spans of their blooms according to each location and the years we had samples for. So you can see on this graph, all the shades of blue represent the blue star on the map, which is Orr's Island. And so we looked at the habitual blooms or annual blooms of Alexandrium. And you can see that the southern sample, circled by the purple circle, their bloom ended about the same time that the northern sample, the yellow circle, their bloom started, which further affirmed our hypothesis that there is a difference. What we also noticed was that the dates for the Alexandrium blooming was so annual. For example, on Orr's Island, Alexandrium was spotted on May 26th on both 2014 and 2013, and previously it had been assumed that blooms were caused by environmental factors such as warmth in the water and high dissolved oxygen levels, but it's not quite known yet. And this kind of I can't think of the word, but it doesn't prove that to be right because of how annual this happened. Environmental factors like that aren't this annual.
For future semesters to go off of our work, we would like to look at environmental data, even though that it hasn't been disproven that environmental data, or environmental like temperature, dissolved oxygen, pH levels in the water isn't what causes Alexandrium fundyense to bloom at such a precise time every single year. It would still have to know what's going on in the water at the same time that Alexandrium fundyense is blooming. So temperature, pH, dissolved oxygen would really help us understand what's going on all at the same time. Another process of our work that we would like to happen in future semesters would be cloning. So what we did was we did whole genome amplification, which amplified the genes that we precisely wanted to look at. But since they weren't isolated, we weren't able to precisely look at the genetic differences between the 2 cisbeds. We would like for the cloning of the whole genome amplification sequences to be amplified and then cloned to isolate a single Alexandrium fundyense cell, so that we can then go through a PCR or polymerase chain reaction one more time and see the genetic differences between the 2 different cisbeds.
We'd like to thank
Firstly, we would like to thank Dr. Carrie Whitaker for her abundance of genetic information and help on this wonderful project that we got to pursue. Secondly, we would like to thank our TA in Marine Science, Maddie Halloran, for her extended amounts of positive support and help on lab reports.
We'd like to thank the Maine Coastal Program for supporting our research and supporting the future research projects to come.
We'd like to thank the Department of Maine Marine Resources for providing us with this opportunity and giving us this project, and Allison Surwa for giving us her passion in fundyense and teaching us about it to begin with.
A great thanks to the volunteers of the Maine DMR who actually collected samples before we even started our project, and Coastal Studies for Girls for being such an amazing environment for us to do science in and for us to live in.
Any questions? Yeah?
So is the idea that the bloom could be something like when you get annual like hay fever, like blooming in a tree, that could just be happening in the ocean? Is that the idea?
Yeah, a bloom is like that. It typically happens in late May to early June, and we don't know what causes it yet, but Alexandrium will not become dormant anymore and rise up from the cyst beds, and they'll like reproduce, and it's a big issue, and then it kind of goes down like pollen does in the fall.
Because of previous research has discovered that because the Alexandrium can stay in cyst beds for up to 100 years, that it's assumed some kind of condition is affecting it so that it only blooms when it's optimal. So it may not be annually, but that's annually has been observed in the Gulf of Maine.
Other questions? All right, let's give them another round.